The use of recombinant proteins in medicine. Today, with the sufficient knowledge in molecular biology of microorganisms, scientists have been able to accelerate the development of improved biological products and processes. Biotechnology has been able to manipulate the functional units of inheritance, the genes. The technique of isolating the genes and transferring them from an organism to another is what we now call recombinant DNA technology. Recombinant proteins are produced from molecular cloning. It's a process of transferring the genetic information, the DNA, from one organism to another. The foreign DNA from an organism is extracted and cleaved off enzymatically, then joined together with another DNA segment called a cloning vector. These cloning vectors can be of plasmids, viral vectors, cosmids, or even artificial chromosomes. This new DNA construct is then transferred and maintained in a host cell. The protein product is then encoded by the cloned DNA inside the host cell. Proteins perform a wide and crucial range of biological functions. They function as catalysts, they transport and store other molecules such as oxygen, they provide mechanical support and immune protection, they generate movement, they transmit nerve impulses, and they control growth and differentiation. Given these important biological functions of proteins, the synthesis of protein through protein recombinant technology has enabled science to isolate DNA and produce proteins like hormones, enzymes, antibodies, and more, and applied this in different fields of molecular biology. For example, in medicine, human diseases can be a result from the dysfunction of some proteins. Therapeutic proteins can treat a variety of diseases like diabetes, cancer, hemophilia, and anemia. Some of the therapeutic proteins include antibodies, hormones, enzymes, and anticoagulant. Another field where recombinant proteins are used is in research to help uncover the functions of other genes in various cellular activities like in cell signaling, in metabolism, in growth, replication, and in cell death. They can help to understand the transcription, translation, and proteins modification processes. Recombinant proteins are also used in food technology, in agriculture, and bioengineering. The history of recombinant proteins. In 1953, a scientific breakthrough which was awarded with a Nobel Prize made by Francis Crick and James Watson for their discovery of the double helix structure of the DNA. Recombinant DNA technology would not have existed without the knowledge of the DNA structure. And in this picture we can see Francis Crick and James Watson with their DNA model. In 1962, John Mathey and Marshall Nirenberg discovered the transfer RNA and that it contains a complementary sequence which base pairs with the template messenger RNA to produce chains of polypeptides of proteins. The first word of genetic code has been discovered. This experiment also showed that messenger RNA controlled the production of specific types of proteins. In 1968, R.W. Hawley was awarded with the Nobel Prize for working out the structure and function of the transfer RNA, the synthesis of protein from messenger RNA, and for cracking the genetic code. In 1969 to 1970, Paul Berg and Peter Loban has conceived the idea in which to create a recombinant DNA in vitro and use them to manipulate gene across species. In 1971, Paul Berg gene splicing experiment of a bacterial virus by the endonuclease restriction enzymes and the joining the DNA has resulted in the first man-made recombinant DNA. In 1972, a study by Mertz and Davis established that using the restriction endonucleases from the E. coli that cut DNA at specific site would create a cohesive ends of the DNA molecules, and that it can be readily recombined, creating a hybrid DNA molecule. At that stage, all experiments were put to halt after Paul Berg experiment to further investigate any potential biohazards and the possible safety and the containment of this new technology. In 1973, Stanley Cohen and Herbert Boyer established the foundation of recombinant DNA biotechnology by providing the experimental basis that plasmid can act as vehicles for maintaining cloned genes. In 1978, the first therapeutic protein produced by recombinant DNA technology was the human insulin. Genentech, which was founded by Cohen and Boyer, has synthesized the human insulin for the treatment of diabetes. The DNA sequence which codes for the human insulin synthesized and transplanted into a plasmid and maintained in the common bacterium E. coli. The bacteria cell has acted as the biological factory for the production of two peptide chains of human insulin. In 1983, Kary Mullis has invented and developed the polymerase chain reaction. The polymerase chain reaction has revolutionized molecular biology. Kary Mullis has won a Nobel Prize for this invention. The PCR is a method to create copies of DNA fragments through repeated cycles of denaturing, annealing, and synthesizing, driven by DNA polymerase. This technique is used in DNA cloning.
The production of recombinant proteins through molecular DNA cloning and protein expression. The procedures are to obtain a donor DNA for cloning. The DNA sequence is extracted and fragmented to a preferred size. This is achieved by the aid of a restriction enzyme called the restriction endonucleases discovered in bacteria. There are many common purified enzymes in manipulation of DNA in vitro, which are used to cut and join DNA to construct recombinant molecules, each play an important role in genetic engineering. The restriction endonuclease has strict specificity for nucleotide sequences and then cleaves the phosphodiester bonds from the sequence. Some of these cuts are staggered symmetrical cuts away from or in the middle of recognition sites, while some cuts are made away from the recognition site. These staggered cuts leave a single strand overhang, sticky ends, at the site of cleavage on the DNA, while some are blunt ends due to cutting in the middle of recognition sites. The unpaired nucleotides will pair with other overhangs, usually any two DNA pieces with the same sequence within the sticky ends can pair, while the ones with blunt ends can be ligated irrespective of the sequence at the ends. The next procedure is to identify a cloning vector to act as a carrier using the same restriction enzyme to cut open the DNA. The vector rings opens up, leaving a space for the donor segment to be inserted, in which case both the complementary ends, the sticky ends of both types of DNA, unite randomly by hydrogen bonds. This is because of the palindromic nature of the restriction enzyme's recognition sites. This happens in a test tube in a solution. Several forms of union are possible: donor-to-donor fragments, vector-to-vector fragments. Most important is donor-to-vector fragment. These recombinant DNA associations are not stable until an enzyme called ligase seals the sugar phosphate backbone covalently linked, forming a continuous and stable double helix. The mixture now contains a population of vectors, each containing different donor. Insert. The solution is then mixed with live bacteria cells host or other known expression vectors that have been treated to make their cells permeable to DNA. Through the process of transformation, the recombinant molecules enter the living cells. Usually only a single recombinant molecule will enter any individual bacteria cell. Once inside the host, the recombinant DNA replicates itself and many copies produced. This original mixture is spread out on a petri dish so that the individual cells are separate from each other, and then they undergo successive rounds of cell division visible to the naked eye. Colonies form. Thus, the Petri dish contains many hundreds of distinct colonies representing many clones of different DNA fragments, referred to as a DNA library. Then selection is made of host cells that contain the recombinant DNA using selectable markers to identify recombinant DNA. Restriction endonuclease enzymes are restriction enzymes that cut the DNA from the target and the cloning vector for molecular cloning to happen. There are various types of cloning vectors. Every vector should have the following characteristics. 1. Origin of replication. This is a characteristic which allows the plasmid to self-replicate and allows it to act as a vector. Number 2. Multiple cloning sites. Cloning site is a joining of vector to target DNA, stretching a region where it can be cut and cleaved with the target DNA. Multiple cloning sites should have a stretch of lipid region whereby using multiple selection enzymes to fragment, E. coli, HIMD3, BAM-HI. This will allow the easy transfer of a DNA fragment from one plasmid to another. Number 3, a selectable marker. Not all the time will the vector DNA and target DNA will join. Therefore, they cannot be inserted into the host. Once adding ligase, some will join vector to vector, self-join without incorporating the target DNA. Donor to donor can also pair, but very rare. The possibility is the recombinant DNA, vector to donor. We only want to select those host cells that carry the recombinant DNA. This is determined by the presence of the selectable marker which carries the antibiotic resistant gene most of the time. We only select those host cells that carry this antibiotic resistant gene. The selectable marker region with overlapping multiple cloning sites which can be cloned. When the gene of interest is inserted, the DNA will inactivate the antibiotic resistant. The function of the antibiotic resistant properties will not be there. For example, if a certain DNA is inserted into this region, it will cause the DNA to grow in the presence of the antibiotic, a positive selection. The type of cloning vectors include plasmids, cosmids, yeast artificial chromosome, retroviral vectors, bacteriophage, bacterial artificial chromosome, and human artificial chromosome. Different cloning strategies used for cloning prokaryotes and eukaryotic genes. Subdividing genomic DNA into elements that can be cloned and inserting them into a host vector is called creating a library. With many genomic sequences made available, it is possible to clone genes without the need to construct a DNA library. The polymerase chain reaction is a molecular biology technique used to make many copies of small sections of DNA or a gene known as amplification. The PCR involves the process of heating, cooling called thermal cycling, which is done by machine. Every cycle doubles the amount of DNA synthesized in the previous cycle. With each round of DNA synthesis, the newly generated fragment serves as templates within a few cycles, the product being a single species of DNA fragment whose length corresponds to the distance between the two original primers. By 20 to 30 cycles, the effective DNA amplification is achieved, with each cycle acting as a DNA template for the next. PCR makes possible cell-free molecular cloning of a DNA fragment in a few hours compared with several days required for standard cloning procedures. Technique: expression vector, bacteria, yeast, insect, mammalian. Bacteria. Enterobacterium E. coli is the most widely used and first choice microorganism expression system for cloning and genetic modification to produce recombinant proteins. Yeast, Saccharomyces cerevisiae. The target protein is not in soluble form in the prokaryote system or a specific post-translational modification, and this is the approach used. Insect. Baculovirus is a eukaryote expression system. Expression of exogenous proteins in this system is the more popular method. Mammalian. Recombinant proteins expressed in mammalian cell systems use plasmid transfection and viral vector infection. Transgenic animals. Using avian and mammals in the production of recombinant proteins poses a biotechnical challenge in terms of safety concerns such as transmission of infectious disease, immunogenic and autoimmune responses is usually secreted into egg whites or milk of the animal. Transgenic plants. Used in research and diagnostics in the production of recombinant proteins, procedures has its advantages in high yield, low cost of cultivation, lack of human pathogens, and in eukaryotic post-translational modification. The first recombinant product was human growth hormone. Now antibodies serves antigen if hepatitis B virus and other products. Main disadvantage is that the plant-specific post-translational modification in recombinant proteins produce adverse immune response. Applications: medicine produces therapeutic product improving health. Agriculture enhancing food resources through genetically modified foods. Energy applications: bioethanol, bioethanol. Diagnosis: gene therapy and monitoring devices. Application of therapeutic recombinant proteins can be classified into 4 groups. Group 1, therapeutic proteins with enzymatic or regulatory activity. Group 2, therapeutic proteins with special targeting activity. Group 3, therapeutic proteins as vaccines. Group 4, therapeutic proteins as diagnostics. Medical use. Enzyme. DNase 1. Cystic fibrosis is a fatal hereditary disease caused by a highly mutant cystic fibrosis gene, making individuals highly susceptible to bacterial infections in the lungs. Antibiotic treatment has led to the selection of antibiotic-resistant bacteria. Scientists at the US biotechnology company Genentech isolated the gene for human enzyme deoxyribonuclease 1 and expressed the gene in Chinese hamster overalls in culture. The purified enzyme being delivered in an aerosol mist to the lungs of cystic fibrosis patients, decreasing viscosity and adhesivity of mucus in the lungs, making it easier to breathe. Hemophilia is a rare inherited bleeding disorder due to mutation of the gene that encodes the coagulation of protein factor VIII. Losses of coagulation factor VIII function results in bleeding disorder. Hemophilia A, which requires FA replacement therapy. HKB1, a hybrid cell line of HEK293 and a human B cell line, is an efficient host cell line for expression of F8 and possibly other recombinant proteins. Monoclonal antibodies. Rituximab, ibrutinib, a monoclonal antibody that treats non-Hodgkin's lymphoma. Malignant growth of B or T cells or lymphatic cells. Rituximab is a chimeric monoclonal antibody directed against the CD20, a protein on the surface of B lymphocytes. Rituximab kills the antibody-marked B cells. Vaccines. Human papillomavirus, commonly cause of common sexually transmitted disease. Gardasil, a quadrivalent vaccine, contains virus-like particles from major L1 proteins, the 4 major types of human papillomavirus. These L1 proteins reassemble into virus-like particles that resemble papillomavirus, which are highly immunogenic. The gene for the L1 protein from each of the 4 viruses was cloned and expressed in recombinant Saccharomyces cerevisiae yeast strain. Hormones. For more than a decade, treatment of diabetes was with animal insulin. In the early 1980s, human insulin was made available using recombinant DNA technology, using the first commercial healthcare product from this technology. Using the chimeric bovine GH, amino acids in human GH amino acid was constructed and expressed in E. coli bacteria. Other approved therapeutic proteins from E. coli include human growth hormone and parathyroid hormones. Applications in gene therapy, treating genetic diseases such as cancer. Gene transfer, introducing a foreign gene directly into a cancer cell or into surrounding tissues. The goal is that the newly inserted gene will cause the cancer cell to die or prevent cancer cells and surrounding tissue from funneling blood to tumours, depriving them of nutrients which they need for survival. There are several obstacles as well, including gene silencing, on which the implanted genes fail to switch on. Applications: gene therapy, genetically engineered viruses. This approach uses modified viruses called oncolytic viruses, leaving only normal cells unharmed. They target and destroy cancer cells. The virus is being engineered to contain certain genes, designed to infect cancer cells and inside produce proteins that destroy cancer cells. Immunotherapy, CAR T cell therapy, which seeks to enhance the natural cancer fighting ability of patients' own T cells. The patient's T cells are combined with a vector of DNA. The genes of the virus causes the T cells to express a certain protein called a chimeric antigen receptor, or CAR, on the surface. The CAR directs a T cell to the tumour and seeks to destroy them. When the cells, now called CAR T cells, are infused into the patient, they proliferate by generating more cancer cells and seek to destroy tumour cells. Another form of immunotherapy involves gene therapy as cancer vaccines. This approach involves collecting tumour cells from a patient and engineering them with genes that cause them to be more conspicuous to the immune system. The altered cells are then reinfused into the patient along with an immune-stimulating compound. The patient's immune system launches a vigorous attack not only on the newly infused cancer cells but also on similar cells throughout the body. Enzyme product therapy. With cancer treatment methods becoming more focused on targeted approaches in order to maximize treatment efficiency while reducing side effects. One of these targeted approaches is termed suicide gene therapy, also known as gene-directed enzyme pro-drug therapy. The success of the suicide gene therapy is dependent on its three major functional components: enzyme, pro-drug, and gene delivery system.
In this part of our presentation, I'm going to discuss with you the advantages and the shortcomings of the techniques in the production of recombinant DNA proteins in medicine, and also the potential future applications. Recombinant DNA technology has many advantages. One of the more evident ones is the major advancements these medicines have provided to the prevention, diagnosis, and treatments for a number of diseases. Since the recombinant form of insulin was produced in the 1980s, there are more than 200 recombinant protein drugs approved to treat a range of diseases such as cancers, multiple sclerosis, cystic fibrosis, strokes, clotting disorders, and a number of other diseases. As well as the advantages of treating a range of diseases, recombinant protein technology generally offers patients greater safety and less invasive techniques in comparison to previous methods, which involve the isolation of therapeutic proteins from their natural sources, and this increased the risk of pathogen transmission to the recipients. Some examples of this is the past treatment of haemophilia, in which anti-haemophilic proteins were isolated from bovine and human blood. As a result of this, there were high incidences of severe allergic reactions, and also there were many cases of HIV and hepatitis C infections, which were transmitted from the viral contaminated blood to the recipients. Even with modern technology, some viruses are resistant to inactivation methods, and therefore recombinant protein therapeutics can offer better patient safety. Another advantage of recombinant protein therapeutic production is the ability to produce large-scale quantities of these proteins. And if these were otherwise sourced in their natural environment, for example from humans, not only would it be invasive, as discussed earlier, but this method would likely only produce very small quantities of these proteins. This is beneficial to the biopharmaceutical industry in terms of cost and efficiencies, and also to the medical industry and to patients to provide greater access to these medicines and therefore contribute towards improved patient well-being. Various techniques used in the production of recombinant protein therapeutics, such as the expression system choice, protein engineering, and downstream modifications of the protein provide the ability to enhance desirable results, such as the yield, stability, solubility, and other functional characteristics. For example, using E. coli as the expression system is a low-cost alternative, is easily manipulated, and is capable of producing high yields of the recombinant proteins. Mammalian cells produce high-quality and complex recombinant proteins due to their cellular compartments providing post-translational modifications. Yeast systems are also capable of performing some of these post-translational modifications. Although there are clear advantages to recombinant protein therapeutic techniques, there are also a number of shortcomings. Proteins are large, complex structures with primary, secondary, tertiary, and even sometimes quaternary structures. They also have reactive side chains which impact the functional characteristics such as folding and interactions with other molecules. It can be challenging for biopharmaceutical companies to create safe, stable, and soluble recombinant protein medicines. And there can be over 5,000 critical steps in the production of recombinant proteins. During production, factors that can cause misfolding and denaturation of the protein, such as proteolytic activities, lack of disulfide bridge formation, and cell stress, can cause the recombinant proteins to aggregate into inclusion bodies. This is generally unwanted because it impacts the protein's solubility and stability, and this may cause a loss of bioactivity and decrease the safety of the therapeutic. Also, further recovery methods are often required to correct the loss of bioactivity, which increases the cost of production. There is also an increased risk of antibody development with recombinant protein engineering and purification techniques, especially when there has been changes to the protein structure in comparison to its natural form. Other challenges to producing safe recombinant protein medicines is overcoming the short half-lives of proteins and the low oral and transdermal bioavailability. This is often combated through creating injectable protein medicines or sustained release formulations. Other shortcomings of producing recombinant protein therapeutics is the cost and legalities for the biopharmaceutical industry. In comparison to chemically synthesized molecule drugs, the cost of recombinant proteins is much higher and relies heavily on investment. Each project can cost around $1 billion, and it is estimated to take approximately 12 years from the initial idea of the recombinant protein right through to the development of a commercial product. There is a greater than 90% risk of losing investment during production, and therefore it is extremely important that inventions are protected from competitors. Patents are a form of intellectual property rights granted by the governments which give inventors the exclusive rights to commercially develop products from the patented product. However, filing patents can be costly and frustrating, and they often need to be filed in multiple countries in order to protect the invention. Once patents have been granted for recombinant proteins, competitors must obtain licenses to develop products from the original, or otherwise change their direction in product development. Even with the challenges to making these biopharmaceuticals, the future looks promising and holds vast possibilities for medicine. The number of recombinant biopharmaceuticals is predicted to grow rapidly and account for around half of all medicines within the next decade. There is a further need for improvement to recombinant protein therapeutic research and production, such as the increased knowledge of protein functionality, the genetic code, best choice of expression system, and protein engineering. This will facilitate the production of new biopharmaceuticals as well as enhance the functionality of existing treatments to increase the safety, efficacy, and reduce the invasiveness of current techniques. In the future, there will be an importance to create recombinant protein medicines for diseases that lack alternative treatments, as there is a waiting market for these. Other future areas of interest are drugs that can cross the blood-brain barrier, monoclonal antibodies, which accounted for almost half of recombinant medicines approved in recent years in the US, as well as targeted therapeutics and antigen-specific antibodies. Other areas of interest are hematology and oncology, which remain the two dominant areas for approved recombinant protein medicines. To improve the functionality, costs, safety, and efficacies of recombinant protein therapeutics, advances to protein engineering and recombinant DNA techniques will likely be a future area of interest. Some brief examples of this are polyethylene glycol attachment or pegylation to recombinant proteins to minimize protein degradation and lower the immunogenicity risk. Also, co-expression with chaperones or foldases to assist correct folding, and thus less downstream modifications will be required, as well as the use of fusion proteins. This can extend the half-lives of the medicines, provide targeting functions, increase the receptor binding affinity, allowing for lower dosage requirements. During production, fusion proteins can facilitate exportation from the culture cells, reducing the costs due to more efficient isolation of the proteins. Thank you for listening to our presentation.